Hello, everyone, and welcome. We're excited to have you here for today's fast 30-minute webinar on using shear flow to study biofilms in realistic environments. Let's be honest, biofilms don't grow in still plates. They grow under flow, under stress, and under conditions that are, let's just say, a little dramatic. And that's exactly what today's session is about. Real-world biofilm behavior powered by flow and real case studies. Just a few quick notes before we start. This session is only 30 minutes. You can drop questions in the Q&A anytime. And yes, we'll send out the recording afterwards. So let's get started. I'm excited to introduce today's speaker, the brilliant Dr. Stephanie Proano, a PhD level scientist with experience spanning government research, GMP-regulated pharmaceutical manufacturing, and biotechnology. In her current role as a field application scientist, Stephanie works closely with researchers, clinicians, and industry teams to translate complex biology into practical real-world workflows, from optimizing experiments to bringing new technologies into daily lab use. With expertise across neuroscience, molecular biology, and cell physiology, and hands-on skills in electrophysiology and live cell imaging, she's uniquely positioned to guide today's conversation on dynamic biofilm research under flow. Stephanie, we're thrilled to have you. Take it away.
Hi, everyone, and thank you so much for joining today's webinar, and thank you, Lisa, for that kind introduction. Today, we'll explore how harnessing shear flow can transform the way we study biofilms. By the end of this session, you'll understand how to use the BioFlex platform to recreate physiologically relevant biofilm microenvironments, how to capture dynamic cellular responses that static cultures often miss, and how to apply real-world BioFlex data to improve our understanding of biofilm behavior and support therapeutic discovery. So what are biofilms? Biofilms are communities of microorganisms embedded in a self-produced extracellular matrix that helps them adhere to surfaces, stick to each other, regulate nutrient and chemical signals, as well as support metabolism and gene expression. Biofilm formation starts with loose attachment, progresses to irreversible adhesion and matrix production, and ends with dispersal that seeds new biofilm communities, beginning the cycle all over again. In human health, biofilms play a major role in many infections, including dental and periodontal disease, respiratory, urinary, and GI infections, chronic wound healing, and infections resulting from medical devices like respirators, catheters, and orthopedic implants. It is estimated by the NIH and the CDC that over 80% of microbial infections involve biofilms, costing the US about $6.5 billion annually. The biggest challenge with combating infections caused by biofilm-forming cells is that they are 10 to 1,000-fold more resistant to antimicrobial treatment than planktonic cells. Biofilms provide the perfect environment for these organisms to evade the host immune response and develop drug-resistant strategies, which contributes to persistent infections and rising antimicrobial resistance, which is estimated currently causes about 700,000 deaths per year, which are projected to rise to 10 million annually by 2050, according to the World Health Organization. Inside the human body, fluid flow, temperature, as well as nutrient and gas gradients are persistent in different compartments, from blood in the circulatory system to saliva flow in the mouth and chyme in the gut, lymph in the lymphatic system, and urine in the renal system. Unfortunately, many research models fail to reproduce these dynamic conditions, limiting our understanding of how biofilms really behave in vivo and hindering the development of effective treatment strategies. Traditional in vitro biofilm models rely mostly on standard static cultures that don't support co-culture or physiological flow, lack control of temperature, gas, or nutrient gradients, and often require complex setups with pumps, tubing, and external regulators. These limitations oversimplify the true biofilm microenvironment. To overcome these limitations, we need experimental platforms that bridge the gap between static culture conditions and the dynamic environments in which biofilms truly form. The BioFlex bridges this gap by offering controlled physiologically relevant shear flow, built-in temperature and gas regulation, real-time live cell imaging, all in a low-maintenance microfluidic operation, which enable more precise, clinically meaningful biofilm experiments. So how does the BioFlux work? The BioFlux controller is a benchtop electropneumatic pump that generates controlled air pressure and provides precise temperature and gas control, and it also drives flow through the microfluidic channels inside BioFlux plates, which are the only components through which fluids flow. BioFlux experiments pair this controlled fluid flow with live imaging to capture biofilm development in real time within the environment of BioFlux plates, which are SBS standard well cassettes with microfluidic flow cells sealed over a 170 micron glass cover slip for high quality imaging. The microfluidics in the plates vary in their architecture and purpose and can support anywhere between 3 to 24 experiments per plate. When doing an experiment, once reagents or cell suspensions are loaded into the plate wells, the plate is placed on an inverted microscope and is sealed with the plate interface that is connected to the BioFlux controller. Air pressure from the controller is delivered through each individual well within the plate, and it is this air pressure that pushes fluid through the microfluidic channels at a defined shear rate. As the experiment unfolds, time-lapse images capture biofilm growth and behavior over minutes to hours. From these images, we can quantify biomass coverage, object counts, as well as fluorescence intensity. Now that we've covered how the BioFlux works, let's look at a few published or peer-reviewed studies that leverage the platform. For the first study, we see how two-phase flow, which allows flow of two fluids within the same microfluidic channel, was used to understand interspecies interactions in an oral biofilm environment. The purpose of the study was to understand the influence of an extracellular DNA enzyme produced by S. gordoni, called streptococcal surface nuclease A, or ssNA, on the colonization capabilities of Streptococcus mutans. To this effect, cell-free filtered saliva was introduced to the microfluidic flow cells of 24-well BioFlux plates at a flow rate of 0.5 dynes per centimeter square for a duration of 18 hours or 37 degrees Celsius to allow biofilm formation. Once the biofilm was established, it was stained with an extracellular DNA stain, and cell-free filter saliva plus ssNA were perfused only on the bottom half of the same microfluidic channel in which the biofilm originally developed. In the figure, we can appreciate that the presence of ssNA caused the breakdown of extracellular DNA released by other bacterial species found in saliva, inhibiting the growth of oral microcosm biofilms in human saliva. In this study, the BioFlux effectively enabled the study of two different conditions, the control condition on the top of the microfluidic channel and the treatment condition on the bottom of the microfluidic channel, on the same biofilm and within the same microfluidic channel under more physiological conditions that mimicked saliva flow in the oral cavity. Next, we see how the BioFlux was leveraged to do anti-biofilm screenings of compounds. Using the 48-well plate, which has a total of 24 microfluidic flow cells, the antibacterial properties of diethyl dithiocarbamate, or DDC, and copper ions were studied on a Staphylococcus aureus biofilm, which is a common pathogen isolated from surgical site infections. DDC was chosen because recent reports have shown that it is effective in combating fungi, parasites, viruses, and bacteria. Therefore, the aim of this study was to assess the antibacterial properties of DDC in combination with copper ions, which together might have enhanced antimicrobial activity. To study this, methicillin-resistant Staphylococcus aureus biofilms of the mu50 strain were grown in the microfluidic cells of BioFlux plates, under constant nutrient flow from tryptic soy broth, alone or in combination with DDC and copper ions, at a flow rate of 0.5 dynes per centimeter square for a duration of 24 hours at 37 degrees Celsius. As we can see in the figure, the treatment with DDC and copper inhibited biofilm formation over a 24-hour period, showcasing the therapeutic potential of this combination treatment and also highlighting the bioflux's ability to run biofilm experiments long-term under constant nutrient delivery. In the following study, we see how the bioflux was used to develop an in vitro model of diabetic foot infections and one of its most common treatments, debridement. A mixed culture of reference and clinical strains of Staphylococcus aureus and Pseudomonas aeruginosa was introduced in the microfluidic channels of 48-well bioflux plates at a flow rate of 0.2 dynes per centimeter square with either brain-heart infusion broth, the control condition, or chronic wound media, which contains three major constituents of the chronic wound bed. It contains red blood cells, serum, and damaged tissues. The biofilm was established over a period of 72 hours at 37 degrees Celsius. Following this time, biofilm debridement, an important step in diabetic foot infection management, was achieved by flowing media at a higher flow rate of 5 dynes per centimeter square in the forward and reverse direction for a couple of times, which reduced the coverage of the biofilm to about 15%, which is represented by these black lines in the figure. After debridement, a panel of antibiotics at 10 times the minimum inhibitory concentration was added to the media and allowed to flow for an additional 24 hours at 0.2 dynes per centimeter square. These antibiotics were tested alone or in combination with each other. The results reveal that debridement alone is not a perfect treatment because it doesn't eliminate biofilms entirely. However, adding antibiotic combination therapy to treatment was effective at further decreasing the amount of biofilm coverage in this in vitro model of the diabetic foot infection. Catheter-associated urinary tract infections are one of the most frequent biofilm-based device-related infections. So in this next study, the aim was to understand the ability of poloxamer 338 to clear or prevent biofilm formation. Poloxamer 338 is a class of copolymer that has gotten recent attention for its cleansing properties and tissue repairing capabilities. For this study, clinical E. coli strains collected from urinary catheters were allowed to grow into biofilms in 48-well bioflux plates. The strains were constantly fed with lysogenic broth supplemented with glucose at a flow rate of 0.5 dynes per centimeter square for 18 hours. The biofilms were then treated with a 0.5% solution containing poloxamer 338 alone or in combination with cefoxitin, an antibiotic commonly used to treat these kinds of infections. Following treatment, a live dead stain was used to determine bacterial viability. And to orient you to the stain, cells that appear in green are live and those that appear in red are dead. From the figure, we can see that the area covered by the EC9 FSL strain biofilm was reduced by about 60% when the combination therapy was used, as opposed to about 25% when only half of the minimum inhibitory concentration of cefoxitin was used. And we can also see that the number of dead cells significantly increased post-treatment. These results not only show that the bioflux can be used to recreate the environment of a urinary catheter, but that poloxamer 338 might be a great therapeutic candidate to eradicate catheter-associated biofilm infections. In the last study that I'll highlight today, H. pylori biofilms were grown in bioflux plates to understand the relationship between antibiotic resistance and the bacteria's ability to form biofilms. 24 clinical strains of H. pylori obtained from gastritis patients were sorted into different categories dependent on their reaction to a host of antibiotics. The bacteria were sorted into antibiotic sensitive, monoresistant, double resistant, and multi-drug resistance groups. After initial screening with crystal violet, 6 strains were chosen to grow biofilms under flow for 24 hours and be further subdivided into either weak or strong biofilm producers. The biofilms were then stained under flow conditions with cyto9, shown in green, to look at biomass availability. They were also stained with DAPI, shown in blue, to look at extracellular DNA, and finally cyper-Ruby to identify extracellular proteins, and that is shown in red. The results reveal that all of these markers were heavily present in the biofilms produced by the H. pylori strains that fell under the strong biofilm producers category, shown in the bottom half of the panel. These particular strains showed a strong correlation with resistance to antibiotics, given that they came from either the double resistant or the multi-drug resistant groups previously identified. So with this study, we can see that the bioflux allowed for a successful establishment of H. pylori biofilms under flow. For these real-world applications, we can see that biofilm infections are a major clinical challenge with limited treatment options. Traditional in vitro models fail to predict therapeutic success, and that advancing biofilm research depends on better models that translate to clinical outcomes. The Bioflux provides a physiologically relevant platform by integrating controlled shear flow, temperature and gas regulation, as well as real-time imaging to capture dynamic responses. If you'd like more information or want to request a demo, please reach out to us online.
All right, if that didn't get your biofilms moving, I don't know what will. We have a quick poll question before we start Q&A that will appear on your screen. And then while you answer that, we can start with our Q&A. So Stephanie, for our first question, what advantages does shear flow in the Bioflux provide when studying biofilm formation, dispersal, and treatment response?
Yeah, that's a great question. The advantage is that with shear flow, we can study biofilms as they happen in vivo of course, when the microorganisms normally in vivo grow under shear flow conditions. So with the bioflux, we can provide that. We can mimic their in vivo environment and then track more physiologically relevant responses.
Okay. Can the biofilm support long-term biofilm experiments and multi-condition studies within the same plate?
Yes, it can. So, with depending on the plate format that's used, we can run different conditions at the same time. And as far as the length of the experiments, they can go anywhere from minutes all the way to, I think I've seen in the literature, up to 72 hours.
Okay. How can we set up cyclic stretch-based breathing assays on lung cells?
So that might be addressed by adjusting the gas composition that's delivered to the experiments. And so normally the bioflux can be connected to external gas sources, and then that would be the gas that is delivered into the wells that will push then the fluid through the microfluidics.
Okay. Can we create long-standing gradient in the bioflux?
We can, depending on the plate format that's used, we can generate gas gradients or gradients with media. We have a plate format that supports two inputs into one microfluidic flow cell, and it is this format that allows for the formation of gradients.
Okay. Most, if not all, of the studies were in singular species biofilms. Does this technology also accommodate for research in multiple microorganism biofilms?
It does, yeah. So, for example, we could create a suspension of cells with multiple organisms, multiple species organisms, or as I mentioned, there is the well plate format that has two inputs to one microfluidic flow cell. So, a cell suspension with some organisms could be loaded, and then on the other input, another cell suspension with other microorganisms can be loaded as well, and both of those can flow into the same microfluidic flow cell, supporting multi-species biofilms.
Okay. Do you have a history of FDA acceptance of findings and data derived from your method?
That is a great question. Most of the what's in the literature is early or preclinical research. So that would be pretty much where the realm of the bioflux is.
Okay, can flow rate be increased from 0.5 dyne centimeters squared up to 25?
So we have two plate formats. We have a low shear that can support flow rates from zero point 0.1 to 20 dynes. And then we also have a high shear plate format that can support flow rates from 1 to 200 dynes. So it'll be a matter of knowing which plate to use.
Okay, how does the Bioflux platform compare to setups with external pump and tubing?
So the best thing about the Bioflux is it's how it's connected and its maintenance. So as opposed to those DIY systems where fluid is moving through the tubing, with the Bioflux, all we move through the tubing is pressurized air. So all of the cell suspensions, the biofilms, the fluids, all of that remains within the plate. And because of that, we don't have to clean the system every time or clean the tubing every time we run an experiment.
Okay, how does Bioflux help with co-culture or multi-species biofilm models that are difficult to maintain in static systems?
Yeah, so that goes back to the question earlier. We have the ability, depending on the plate format, we can load with the two inputs, we could load different multi-species cell suspensions, load them in into one microfluidic flow cell, or we could there are many ways to do this. We could create a cell suspension with the different species, or another option would be to flow into the microfluidic channels one species, let that settle, and then load another species into the microfluidic channel and watch those two or multi-species biofilms interact.
Okay, can we use biofilm system used to investigate interaction with anti-fouling surfaces within biofilm or with biofilm?
That we would have to, it depends. The biofilm, sorry, the bioflux, the plates, the bottom of the plate is a glass cover slip, and this can be coated with different matrices or different coatings, and that's one way to adjust the surface on which the biofilm develops. The other option is also to, instead of having the cover slip at the bottom, we can also put in a custom substrate. But we'll have to ensure that the custom substrate is clear enough so that we can still acquire the live cell imaging.
Okay. Are you continuing to improve on the creation, the environments, especially in the surgical environment, where current treatments are ineffective at reaching where nastiest of biofilms?
So if I understand the question correctly, this is to address the biofilms or that could occur due to surgery. And that's certainly an application of the instrument. And it would be a matter of isolating, which has been done in the literature, in which isolates are of bacteria uh, are received from patients, and then these are pushed through the microfluidics, and then the biofilm is controlled over time to see how it grows, and then it's challenged with different antimicrobial reagents.
Okay. Is your clinical biofilm method applicable to environmental research like monitoring biofilms in wastewater treatment plants?
That could also be an application, certainly. Of course, we would have to adapt those experiments to what would happen in a microfluidic flow cell within the plate, but that is also an application. Again, it's a matter of getting the isolates and then trying to simulate the conditions that would be present in wastewater systems.
Okay. Are there limitations to the substrates that can be added to the plate to study biofilm formation? For example, disinfectants, essential oils, diluted electrolytes, etc.
The only limitation to the substrates or the fluids that are added, of course, whatever is added, because it has to be pushed through the microfluidics, it has to be, first of all, a fluid, and then it shouldn't be too viscous of a substrate, because again, we are using air to push the fluid through the microfluidic channels.
Okay. Is it possible to obtain reliable biofilm thickness measurements directly?
So that will depend on the imaging system that's paired with the bioflugs. So if a confocal, for example, is used, it is certainly possible to obtain these measurements.
Okay, it looks like that's all the questions we had. We had a lot of great questions in there. Um, so Stephanie, I'd like to thank you for such an engaging and practical session. And thank you to everyone who joined us today. We'll be sending out the recording and resources shortly. Please do me a favor and take a moment to answer a few questions as you exit the webinar. Enjoy the rest of your day, and may your flow always be smooth and your biofilm always be well behaved.